कौन से बंदे कौन है? माइक्रोबायोलॉजी अथॉरिटी यहाँ पे, ठीक है सर। हाँ, आवाज नहीं आ रही आपकी?
खोले में दिख रहे हैं क्या?
कार्ड में दिखना चाहिए वैसे हम लोग को।
नहीं दिख रहा है क्या?
हमको वो बुलाना चाहिए, बुलाना हमको पता चलता है।
ये तो बोल नहीं होगा, खोलने में दिखेगा। मुझे नहीं पता मैं कितनी रेंज में देखती हूँ इसमें। सर आपका खत्म हो जाए ना तो मेरा वाला चला देना बस, मेरे को इंट्रोड्यूस भी कर देना।
तुम एक बार वो यूट्यूब उधर चला के चेक कर लो क्या। Good morning, everyone. For today's journal club, I am the preceptor and Dr. Jaya Biswas is the presenter. The topic for today's journal club is Heterologous boost flowing Mycobacterium Bovis BCG vaccine reduces the less persistent rather than the early stages of intranasal tuberculosis challenge infection. This article has been taken from the Department of Pathology, Pathogen Biology, and the various institutes from the China, and was published by the Wu et al. And from now I am coming from the now I am coming on the need for the TB vaccines. The need for TB vaccine is so the globally in 2017, 10.4 million new TB cases were detected, and among them 1.6 million deaths are reported, which is the serious issue worldwide. That's why, and the emergence of extreme and totally drug resistant strain are the other serious condition. And among these, approximately 1.7 billion people, which are 23% of the world populations, have the latent TB infection. And globally, 50 million individuals who are latently infected with multi-drug resistant TB. This is the in-TB strategy where the WHO to reduce the TB morbidity by 90% and TB mortality by 95% by 2035. This is the other issues are the increase in co-infection with HIV infection, and there is the other issue like cost of the complete treatment course is matters. That's why the TB vaccine is the most essential tool for diminishing this TB infection. And now I start with the initiation of vaccine response. Following following the following the injection of vaccine, there is pathogen associated pattern which is contained in the vaccine antigen attracts the dendritic cell, macrophages, monocytes, and neutrophils. These dendritic cells, monocytes, neutrophils, which are which helps in which patrols throughout the body or helps in just engulf the Mycobacterium bacilli. And after that, there is the production of danger signals, which activates monocytes and dendritic cells. This activation of danger signals changes their surface receptors. The surface receptors like toll-like receptors, N-formyl methionine receptors, mannose receptors, and scavenger receptors. This induces the migration along the lymphatic vessels to the lymphatic drain lymph nodes, where the activation of the T lymphocytes and B lymphocytes take place, which taking in the part of the vaccine response. And now coming to the response to the protein antigens. This slide shows about the extrafollicular and germinal center response to the protein antigen. Here, when the protein antigen reaching the lymph node or spleen, then the B cells binds to this antigen with their surface immunoglobulins. In and after that, in extracellular reactions, B cells rapidly differentiate in the plasma cells, which produce the low affinity antibodies, which appear as low level in the serum within few days after the immunization. And here, antigen specific T helper cell is also activated by the antigen bearing dendritic cell, which triggers some antigen specific B cells which migrate towards the follicular dendritic cell and initiating the germinal center reaction. This germinal center reaction, B cells receive the signal from the follicular B cells, follicular T cells, and which undergoes the clonal proliferation and switching the IgM antibody towards the IgG antibodies, IgA antibodies, or IgE antibodies, and differentiate into the plasma cells, which secrete large amount of antibodies. And at the end of the germinal center reaction, there is few plasma cells which exits from the lymph node or spleen and migrate to the bone marrow where they survive, and later they are used when the body is infected by the tubercular bacilli again. This is the slide which shows about the response to the polysaccharide antigen. This slide shows about extrafollicular B cells response to the polysaccharide antigen. Here, by using their specific immunoglobulin surface receptors, B cells bind to the repetitive structures of the polysaccharide, which reaches the marginal zone of the spleen or lymph node. Here, in the absence of antigen-specific T cell, B cells are activated and which proliferate in the plasma cells without undergoing affinity maturation in the germinal center. These plasma cells migrate towards the these plasma cells migrate toward the red pulp of the spleen, and here they survive for few weeks or month, and secreting the low level of immunoglobulin M, G, and A antibodies. Now I am coming to the B cell memory response. Memory B cells generated in response to the T cell dependent antigens. During germinal center reaction, parallel to plasma cells, they exist from the germinal center. B cell not differentiate into antibody plasma, but into memory B cells. They persist as a resting cells until the unexposed to the response to the specific antigen. On secondary antigen exposure, memory B cells proliferate and differentiate into plasma cells. And after differentiating into plasma cells, they secrete a large amount of high affinity antibodies within few days after boosting. And now I am coming to the T cell effector response. The T cells, the role of the T cells are much higher than the B cells in the TB vaccine response. Here the antigens, mainly the antigens are phagocytized by the dendritic cell, and then these antigens are processed into small peptides and displayed in the groups of the major histocompatibility complex class 1 and class 2 molecule. Then after that, CD4 T cells with appropriate MSC peptide activated. After this activation, this provides signals to the dendritic cell and differentiate in the effector cell, which produces Th1 and Th2 helper cytokines. The CD4 helper T cells support the CD8 T cells differentiation, which inhibited by the Th2 like cytokines, and then the CD8 cytotoxic cells T cells recognize MSC class 1 peptide complex and differentiate into cytotoxic effector cells, which help in the killing of the Mycobacterium tuberculosis bacilli. And now coming into vaccine response. Vaccine response, the B lymphocytes, which mainly prevents the infection by clearing the extracellular pathogen, by neutralizing viral replication, promoting opsonophagocytosis of the extracellular bacteria. It also activates complement cascade processes, and also it helps to after binding to the enzymatic site of the toxin and their diffusion. And now this is the CD8 plus T cells, which do not prevent the infection but help in the reducing, control, and clearing the extracellular pathogens by their cytotoxic effect. It helps in killing by directly killing infected cell by releasing the perforin, granzyme, etc. and etc. This is our that killing the infected cell indirectly also by the producing the antimicrobial cytokine release. Now I am coming to the vaccine response by the CD4 helper T cells, which help in the reduction, control, as well as clearance of both extracellular and intracellular pathogens. The main subsets or main cytokines or inflammatory mediators which are used in this response is the follicular helper T cells, which secretes interleukin 21, T helper cell effector cells, which secretes interferon gamma, tumor necrotic factor alpha, interleukin 2, which protects against the intracellular pathogen. T helper cell 2 effector cells which secrete the interleukin 4, 5, interleukin 13, which responds to the extracellular pathogen. And the last is the TS17 effector cells, which secretes interleukin 17, which secretes interleukin 17, 22, and interleukin 26, which helps in the contribute in the mucosal clearance of the Mycobacterium tuberculosis. Now I am coming to the immunology of the tuberculosis infection. Tuberculosis. The tuberculosis infection is divided into three stages mainly. The first stage is there is the infection of tuberculosis at the young age by air droplet inhalation. Then in this case, the this includes, actually there is the generation of acquired immunity, which includes the MSC2 restricted CD4 T cells and MSC1 restricted CD8 T cells. Here B cells also activated, but their protective role in TB remains less. At that stage, here pre-exposure vaccines are given to prevent the TB infection. And the other stage is the stage 2. Here the acquired immunity is comprising of CD4 and CD8 plus T cells, which contains Mycobacterium tuberculosis bacilli in a dormant stage within solid granuloma. Here T cells produce type 1 cytokines and cytotoxic effector molecules, and after that they become memory T cells, which produce multiple cytokines, and the individual remains latently infected here without clinical signs and symptoms. In this case, post-exposure vaccines are given to the adolescents or adults who are latently infected but healthy individual. And the stage 3 is the mechanism leading to the deficient immunity and disease reactivation. And there is the production of suppressive cytokines, which is such as interleukin 10 and tumor necrotic factor beta by T helper cell 2 and regulatory T cell. And here the Mycobacterium tuberculosis becomes metabolically active, and there is the formation of granuloma, caseous granuloma. Here therapeutic vaccines are given in addition to the chemotherapy. Now I am coming to the BCG vaccine. The BCG vaccine are discovered by two French scientists. One is the Calmette, a physician, and a Guerin, a veterinarian, who cultured the Mycobacterium bovis in medium with glycerol, potassium slice, and beef wine. And they subcultured every 3 weeks for 13 years. And this M. bovis is from a cow with tuberculous mastitis infection. And the first BCG vaccine is given to human orally in 1921. In India, freeze-dried vaccine, the strain is Danish 1331, is available from the BCG laboratory, which is situated in Gundi, Madras, and the dose is given is 0.05 ml for newborn and 0.1 ml for the older children intradermally. These are the various strains, 5 strains, which are more than 90% of the vaccine used worldwide. Here the Pasteur 1173P2, Danish 1331, Glaxo 1077, and the other one. Now I am coming for what is the need for the new vaccine. The need for new vaccine is mainly due to there is the difference in vaccine efficacy of the BCG, which is highly variable, and in adolescent age there is the waning of the immune memory system, and now the BCG is not recommended in the infants with HIV infection due to the risk of disseminated BCG disease, and if the person is associated with helminthic infection, there is the reduced BCG vaccine efficacy. This is the major problems with BCG vaccine. That's why the new vaccines are needed. Now I am coming to the different phases of vaccine trial. First, it started with the research and preclinical development. Here we identify the relevant antigens and creating concept and evaluation in the animal group. In the phase 1 clinical trial, which started with from the human being, here we conclude the safety in human and immunogenicity. There is less than 100 volunteer and the total duration is 1 year. Phase 2 clinical trial is the preliminary efficacy is detected, and here the dosage and the schedule are also determined. Here the total volunteer is 100 to 1000 and 2 or more years duration are enough. Now, in the phase 3, side effect and efficacy are determined, and the total duration up to 3 to 5 years. And phase 4 clinical trial is the post-marketing surveillance. Here the total duration is 4 to 6 years, and after that the vaccine is coming into the market. These are the pathway for TB vaccine development, which I am just explained in the previous slide, where the preclinical trial, then the clinical trial. Here the various models, here the various animal models are used, like most efficiently animal models which are used is the mice model, guinea pig model, rabbit model, non-primates like rhesus macaw and Cynomolgus macaw monkey, and the other like zebrafish. This type of animals are used for the animal model for vaccine development. These are the pipeline for TB vaccine development. In the phase 3 trial, there is various vaccines like VPM 1002 and VPM 1002 is given, and in phase 2B trial there is MBA 85A antigen vaccine, and the other vaccine associated with adenovirus is also used here. These are the some of the immunization strategy of the current vaccine TB vaccine candidates. It's divided into three types. One is the prime vaccine, second one is the prime boost, and the last is the immunotherapeutic. Prime vaccine is the vaccine which is the replace BCG with either live recombinant BCG vaccine or genetically attenuated Mycobacterium tuberculosis vaccine that provides greater safety and protective efficacy. Prime boost means boost which is limited to the immunity conferred by the BCG vaccine. And here we use either the viral vector or the adjuvant subunit vaccines. It administered as a booster to a BCG prime in infancy and in adolescent when BCG protection is subsided. The last one is the immunotherapeutic vaccine. Immunotherapeutic vaccine is the developed therapeutic vaccine that synergize or with chemotherapy and given with the drugs to shorten the treatment for active tuberculosis disease or for latent tuberculosis infection. It includes the whole cell and fragmented Mycobacterium vaccines. Now coming to the categories of the TB vaccine. Categories of TB vaccine divided into three categories. First is the preventive pre-exposure vaccines. It is given prior to the first exposure to the Mycobacterium tuberculosis in neonates, also known as the priming vaccine. And the second one is the preventive post-exposure vaccine, which is targeted at the adolescent and adult with the latent tuberculosis infection and prior BCG immunization. It is also known as the boosting vaccine. And the last one is the therapeutic vaccine. Therapeutic vaccine is given along with the chemotherapy. And to the person who is at a higher risk of developing recurrent diseases. Now coming to this is the various type of antigen which are used for the subunit TB vaccine. This one is the M72, H1, H4, adenovirus, the 5 antigen 85 here, modified vaccinia ankara 85 vaccine, adenovirus 35 vaccine. These are the various antigens which is used for the development of the subunit TB vaccines. And the other is the adjuvants. These are the various adjuvants like IC31, CF01, GLASC, and these are used, and these are coming in the various phase trials. These are mentioned on the right corner, and these are some virus vectors which are used for the development of TB vaccines are the modified vaccinia ankara virus, adenovirus 5, adenovirus 35, chimpanzee adenovirus, and the flu, which is the replication deficient influenza virus vaccine. And the I am coming to the challenges in TB vaccine development. Preclinical challenges are, this is the unknown correlation between the animal and human protection, and the other one is the lack of immunologic marker of protection. Clinical problems is the protective immunity is not well understood, and the cell mediated immunity induction is very difficult. Uh, there is very large sample size required for phase 3 efficacy study, and there is limited number of field sites for efficacy study. These are the various limitations, various challenges which are associated with TB vaccine development. Now coming to the characteristics property of ideal TB vaccine. The main characteristics of the ideal TB vaccine are it will be safer, protection in animal models. It protects against the disease and infection, and protective after it provides protection after a single or small number of doses. Confer a lifelong immunity and not compromised with the tuberculin test, having in, inexpensive, heat stable, longer shelf life. And these are some of the articles which are published. This article is published in 2015, which shows the immunogenicity and protective efficacy of the DMT liposomal adjuvant tuberculosis subunit CTT3H vaccine. The other article is published in 6 July 2017, and it shows about the multi-stage subunit vaccine effectively, which protects against the mice, primarily progressive tuberculosis, latency, and reactivation. Now, I would like to invite Dr. Jaya for presenting the proper article.
Good morning, everyone.
देखो उसको बैंक का भी ऑन करना था ना, ऑन ही होगा.
Good morning, everyone. I am Dr. Jaya Vishwas, presenting the article proper. Thank you, Dr. Saurabh. My title of my article is Heterologous boost following Mycobacterium bovis (BCG) reduces the late persistence rather than the early stage of intranasal tuberculosis challenge infection. This study was done by Yaki Wu et al. In China, Wuhan. In this study, according to WHO 2017, there were 10 million new TB cases, in which 90% were adult and 10% were children. 1/3 of the world population is estimated to have latent TB infection, of which 5 to 10% would progress to active TB during their lifetime. BCG is recommended to vaccinate infants worldwide and can provide effective protection against several serious forms of TB, and its protection wanes with time. It lasts only for 10 to 15 years. BCG vaccine cannot control the prevalence and transmission of adult TB, mostly pulmonary TB. Therefore, there is an urgent need to develop an effective vaccine against adult TB. So here the vaccine candidates are, which were explored extensively for adult TB. These are DNA and subunit vaccines, recombinant viral vectors, genetically modified BCG, and attenuated Mycobacterium tuberculosis strain. Here in this study, they have used heterologous boost strategy, which aimed to extend the protection period primed by the BCG vaccine. Some study reported that a regimen of BCG prime and adjuvanted multi-stage subunit protein showed a stronger ability to protect mice against M. tuberculosis infection. Therefore, whether heterologous boost regimens are more efficacious to prevent against LTBI than primary infection remains to be investigated. Here is the heterologous boost strategy, which means different type of vaccine given in repetitive immunization with same antigen. And homologous boost vaccine is same vaccine given in the earlier priming immunization. So example of homologous boost vaccine are DPT, tetanus, OPV. But heterologous boost vaccines are recently being studied in TB, HIV and malaria. These heterologous boost vaccines are more immunogenic, increases effectiveness of existing vaccine and easy practical application. So in this study, they have studied 3 CTT3H based vaccines which were constructed. Firstly, DMT adjuvanted CTT3H subunit vaccine. Second is DNA vaccine, which is called PCTT3H DMT. And third vaccine is recombinant adenovirus RAD-CTT3H vaccine. CTT3H vaccine is basically a combination of 5 antigens with CD8 epitopes, which were constructed to increase more immunogenicity. So CTT3H vaccine were efficacious or not, or their protective efficacy and immunogenicity of BCG prime CTT3H based booster were compared in C57BL-C mice of primary and latent TB infection. In this study, they have first constructed these 3 vaccines: recombinant adenovirus vaccine, PCTT3H DNA vaccine, and the DMT adjuvanted PCTT3H vaccine. After that, these mice were immunized with these 3 vaccines and then challenged with M. tuberculosis S37RV strain. After that, they have actually assessed the IgG antibody and their subclasses titered by ELISA. They have also assessed interferon gamma secreted by splenocyte, and they have measured the cytotoxic T cell responses regarding these TB antigens, and CTT3H-specific memory T cell were detected by intracellular flow cytometry. In experiment 1, first they constructed and identified the recombinant adenovirus RAD-CTT3H and eukaryotic expression plasmid PCTT3H and preparation of vaccines. First, the construction of DMT adjuvanted CTT3H subunit protein vaccine. In these vaccines, coding sequence of the subunit antigen, which is consisting of CFP10, TB10.4, TB8.4, RB3615C and HBHA antigens of Mycobacterium tuberculosis S37RV. These coding sequences were amplified by PCR from recombinant plasmid, and these PCR products digested and inserted into corresponding site of the expression vector, and this resultant recombinant prokaryotic expression plasmid transformed into E. coli. So expression of this CTT3H was induced, and this protein was then confirmed by Western blotting. So, and this CTT3H antigen protein was specified by using different monoclonal antibodies, and after these production of CTT3H protein, it was combined with DMT liposomal adjuvant, and formation of vaccine was there. DMT liposome is a chemical substance which is made up of DDA, MPL and TTF, in which DDA is dimethyl dioctadecyl ammonium and MPL is monophosphoryl lipid A and TDB is trihalose 6,6 dihebinate. This DMT liposome was used to increase the immunogenicity. Then they constructed the next vaccine, which is DNA vaccine CTT3H and CTT3H recombinant adenovirus vaccine. In this adenovirus vaccine, they have taken two plasmids. First PDC316 CTT3H and next one is plasmid BH-glox delta E1 33, and these were co-transfected to 293 HEC T cells, in which site-specific recombination was there, and production of recombinant adenovirus is there. In DNA vaccine, they have produced recombinant eukaryotic expression plasmid p-CTT3H, in which CTT3H gene for CTT3H is there. After production of these three vaccines, they have immunized these mice with these three vaccines and challenged with virulent Mycobacterium tuberculosis H37RV. Specific pathogen-free C57BLC female mice were taken, aged 6 to 8 weeks, and they were randomly divided into different groups. And these groups had 6 mice in them. 6 mice in them. So different regimens for these mice were used. Route and doses for these regimens are: first, DNA vaccine, which were given intramuscularly. The dose of this vaccine is 200 microliter twice at 3 weeks, and the negative control for this vaccine is taken PVAX1. And CTT3H DMT, which is subunit vaccine, it is given intramuscularly twice at 3 weeks apart. It is also given 200 microliter of dose. The negative control for CTT3H subunit vaccine, DMT were taken as negative control. For CTT3H, which is recombinant adenovirus vaccine, it is given intranasally. It's given once, 5 into 10 to the power 8 plaque forming unit. The negative control for this vaccine is adrenal, which is also a recombinant adenovirus vector, in which no transgene is present. And in adenovirus vector, there is there is at even place there is CMB polymerase gene, and then this adrenal has not have any transgene. There is also simian virus 40 polyase sequence in place of E1 gene. This is used as gene therapy also. For BCG, which is taken as which is given subcutaneously, the dose of BCG is 1 into 10 to the power 6 CFU. The negative control for BCG is phosphate buffered saline. And this BCG with phosphate buffer saline, it is taken as positive control. So positive control may they taken BCG. For negative control, they have taken PVAX, DMT, adrenal and PBS. After immunizing these mice, they have challenged it with M. tuberculosis strain H37RV. Vaccinated mice were infected intranasally with 100 colony-forming units like Mycobacterium tuberculosis H37 vaccine. Post-challenge, mice were sacrificed and bacterial load per organ was enumerated. For this, first they have taken 3 regimens. In this experiment, for the evaluation of protective efficacy of different type of CTT3H based vaccine against primary M. tuberculosis infection. So they have checked for protective efficacy in primary M. tuberculosis infection. In this regimen, first the group of mice were immunized at first with BCG only, these 3 test vaccine and the negative control vaccine PBS, DMT, adrenal and PVAX. After 1 week, they have challenged these mice intranasally with Mycobacterium tuberculosis. After this challenge, after the infection, 4 weeks later, they have checked for protection, which is done by bacterial load in the lungs and spleen. So in these two graphs, they have assessed the bacterial load with negative control, with test vaccines, and with positive control BCG. So on the y-axis, they have assessed log value, which is log value with colony forming unit per unit lung. Here with negative control, the bacterial load is highest, but with BCG only there is significant reduction in the bacterial load with BCG, and there is also significant load reduction in the test vaccines. The similar result is also seen with spleen. Here the negative control is showing higher bacterial load, and with BCG and this test vaccine they are showing less bacterial load, which is significant. In next regimen, they have assessed the effect of booster vaccine type on BCG prime protection against primary infection. Here they have first primed these mice with BCG at first week. Then they have given single PVS one, BCG one, and this test vaccine and the negative control vaccine. So they have boosted these mice with this vaccine, heterologous vaccine. So after 18 weeks, intranasally they were challenged with TB bacilli. After infection, at 22 weeks, after 4 weeks of infection, they have assessed the protection of these vaccines. So in these graphs, they have seen the bacterial load with negative control, with test vaccine and repeat BCG vaccine. So in the lung, they have found that bacterial load is higher with BCG repeat vaccine, but there is no significant changes with control vaccine and test vaccine. Also, similar results were seen with spleen. Here, this negative control and this test vaccine has similar effect with bacterial load, but BCG repeat vaccine has higher bacterial load. In third regimen, they have assessed the protective efficacy of heterologous boost strategy against latent TB infection in BCG prime mice. So here they taken group of mice in which first they immunize these mice with BCG, and then they challenge these mice with intranasally Mycobacterium. At 5th week they have challenged these mice intranasally. At this point of the time, these mice have 10 to the power 4 and 10 to the power 3.5 CFU Mycobacterium tuberculosis in their lungs and spleen. So this is actually simulating the latent TB infection in these mice. After the challenge, they have given BCG, then test vaccine and the negative control vaccine. After this booster vaccination at 19 week, they have assessed the bacterial load in lung and the spleen. So here at the y-axis they have assessed the log 10 CFU per unit of lung, and they have also assessed the log value of CFU per spleen. Here at the 19th week, with negative control, bacterial load is higher, but with test vaccine, there is significant reduction in the bacterial load, and also repeated BCG vaccine is also having the lower bacterial load than the control vaccine. This similar reaction is also seen with spleen. Here the negative control is showing higher bacterial load, but these test vaccines are showing significant lower bacterial load, and BCG repeat vaccination is also showing the lower bacterial load than the control vaccination. In third experiment, they have assessed the CTT3H-specific IgG antibodies and their subunits IgG1, IgG2A, and their titers IgG1 by IgG2A. Here 1, 2 and A, B, C and D graph is showing IgG titer, IgG2A titer, IgG1 titer, and here IgG2A by IgG1 ratio. They have also assessed the CTT3H-specific interferon gamma secreted by splenocyte detection by ELISA. So in the next graph, in E graph, they have assessed interferon gamma which is secreted by splenocyte. And in this graph, they have assessed the rate of specific killing, which is in vivo measurement of cytotoxic T cell responses to TB 10.4. So they have compared the CTT3H specific Th1 response among the 3 CTT3H based vaccines immunized in C57BL6 mice. So on y-axis they have IgG titer, IgG2A titer, IgG2A titer, IgG1 titer, and IgG2A by IgG1 titer. And on x-axis they have assessed negative control vaccine, the test vaccine, and the BCG positive control vaccine. In this graph, they are showing with negative control vaccine there is no production of or less production of IgG titers, but with test vaccine there is significant IgG titer production, and there is also production of IgG titer with BCG. Similar reaction is seen with this graph also. So there is IgG titer production with the test vaccine in all 4 graphs. And in E graph, this is showing the interferon gamma secreted by splenocyte. With negative control, there is no secretion of interferon gamma, but with test vaccine, there is secretion of interferon gamma. It is greater than BCG vaccine. In F vaccine, they are assessing the cytotoxic T lymphocyte cell count, which is also represents rate of specific killing. Here this is showing with negative control there is no production of cytotoxic T lymphocyte, but with test vaccine there is significant production of cytotoxic T lymphocyte. It is greater than BCG also. Next, in next experiment, they have assessed the TH response in BCG prime CTT3H based boosters. Here, they have assessed IgG titer, IgG2 titer, IgG1 titer, and IgG2A by IgG1 ratio. Here, in second element, here they have actually primed the mice with BCG and then give the negative control. So, this is the negative control. These are the BCG prime boosters test vaccines, and this is the BCG repeat vaccine. Here in all the 4 graphs they have found the IgG type. Here all the 4 graphs they have found IgG titers with negative control also, with test vaccine and BCG vaccine. In these 4 graphs, the highest interferon gamma titer or highest IgG and IgG2A and IgG1 titers are produced with BCG CTT3H DMT vaccine, and interferon gamma is produced by splenocyte. In this graph, they are showing interferon gamma production, and on x-axis there is negative control test vaccines and BCG repeat vaccine. Here with negative control vaccine there is no production of interferon gamma, but with test vaccine there is production of interferon gamma, then the BCG repeat vaccine. And there is also rate of specific killing, which is also represent the cytotoxic T lymphocyte count. Here the cytotoxic T lymphocyte count is also raised with test vaccine. In next experiment, they have counted the memory T cell detected by intracellular flow cytometry. In this experiment, they have counted the BCG. They have counted T cell specific in spleen before exposure of the Mycobacterium tuberculosis. They have counted the CTT3H specific T cells after exposure, and they have counted the memory T cell in lungs after exposure. In this graph, On y-axis, this is showing interferon gamma inducing T effector memory cells. On x-axis, CD4 and CD8 cells. Or here on y-axis, it is showing interleukin 2 expressing T central memory cells, and on y-axis CD4 and CD8 cells. Here they are comparing negative control, which is BCG-PS, and the positive BCG-PCG repeated control. In this graph, they have actually compared the negative control with the test vaccine and the booster vaccine. Here also they compared the three vaccines in these graphs. CTT3H-DMT subunit vaccine, here plasmid CTT3H-DMT vaccine, and here the recombinant adenovirus vaccine. In this graph, in first graph, there is no significant difference with the production of interferon gamma T EM cells and interleukin 2 CM cells with BCG control group and BCG repeat group. But in this graph, before exposure, spleen is producing interferon gamma T EM cell with the CTT3H DMT only vaccine, or it is also producing interleukin 2 expressing TCM cells with CTT3H DMT vaccine. In next graph, they have also assessed the PCT3H DMT vaccine, which is DNA vaccine. In this graph also, these biomarkers are produced by DNA vaccine only, greater than the booster vaccine. In last graph, they have counted the interferon gamma TEM cell with booster vaccine. So here these graphs are showing that CT3H, both three vaccines, which is CTT3H based vaccine. These vaccines are actually producing interferon gamma TEM cells with this CTT3H based heterologous boost induced less significant antibodies than memory T cells than the CTT3H DMT vaccine before exposure in spleen. CTT3H antigen specific T cell in spleens induced by BCG prime CTT3H based boosters after exposure. In this graph, they have assessed the interferon gamma producing TEM cells and interleukin 2 producing TCM cells with the negative control, with the BCG booster test vaccine and the repeat vaccine. And in this graph, they have compared the negative control, boost vaccine, test vaccine and BCG repeat vaccine after the exposure in the spleen. So, among all groups, repeat BCG vaccine induced the most significant increase in the number of TCM and TEM cells, and CTT3H based heterologous boost induced a more significant increase of all three biomarkers than BCG previous control. In next graph, they have assessed the TEM cell and TCM cell in the lungs after exposure induced by BCG prime CTT3H based booster. In this graph, interferon gamma producing TEM cell and interleukin 2 producing TCM cell were expressed mainly with booster test vaccine in all these three vaccines. So, compared with BCG PBS control group, repeated BCG induced a significant increase in biomarkers. So, heterologous booster induced the most number of all biomarkers than the BCG PBS control group as well as BCG repeat vaccination. In fifth experiment, they have compared the histopathological changes in the lungs among different regimens. So here these are lung pathologies which is mainly characterized by vasculitis, alveolitis and fibrosis, and they were given some lung pathological score on the basis of these pathological changes and the acid fast bacilli count. With negative control, with this negative control, PBS, DMT, PVAX and adrenal, the score of lung pathology was highest. In these four conditions, negative control condition, the score of lung pathology was highest, but with test vaccine, these lung pathologies were lower, or with BCG it is the lowest. So this is actually showing that these vaccines are effective. In next slide, this slide represents the lung pathology changes on lung pathology changes after BCG prime CTT3H based boosters against the mice in the late persistent TB. Here in negative control vaccination, the score of lung pathology is higher than the booster vaccine, test vaccine. So to conclude, the results indicate that BCG prime boost strategy might be promising measure for the prevention against the late persistent TB infection by induction of interferon gamma TEM cell and interleukin 2 expressing TCM cell in the slum. It can be used as alternative biomarkers for guiding the clinical practice and future development of TB vaccines. Similar protective efficacy against early intranasal infection was provided by different CTT3H based vaccine alone in vaccinated mice, and their protection was inferior to that of the BCG vaccine. So, CTT3H based heterologous booster did not enhance the protection confirmed by the BCG vaccine against primary infection. All of these three boosters provided stronger protection against late persistent TB infection than the BCG alone. So, regardless of the vaccine type. The limitation of this study is to that these results might not simulate in the humans also. So, a specific studies to be done in humans. Thank you.
Any question? Not a question. हमने critical appraisal नहीं बनाया था.
Okay.
Off कर दिया.
Where is the question? Why there was no protection against primary infection?